Today we are going to isolate the fungi from the disease specimen. It's a rose leaf which have the leaf spots on it, and we are interested in the pathogen which is deep seated inside the tissue of the rose leaf. We will wash this leaf to remove all the dirt having surface borne pathogens on it. Disinfectant. For different plant parts, we use different concentration of the sodium hypochlorite. It is generally known as Clorox. For the purpose of sterilization of the tender leaves, we will use 2% solution of sodium hypochlorite. And this solution, sodium hypochlorite solution, it is 12% solution. So in order to make the 2% solution out of 12% solution, we will use the formula C1V1 is equal to C2V2. C1 is the concentration of the sodium hypochlorite, that is 12%, and how much volume of this sodium hypochlorite we are going to use to make the volume 10 ml, and the concentration which we require is 2%. So what will we do? We will divide it with the 12, and it will be 20 divided by 12, which is equal to 1.6 ml. So this much volume will be used to make 10 ml 2% solution of this sodium hypochlorite, 10 ml volume of sodium hypochlorite. How much volume will we take of the water? .4. So 8.4 ml water, sterilized distilled water rather, plus 1.6 ml 12% sodium hypochlorite, it will make 10 ml. We will label it. After our sample has been dried, we will bring it into the laminar flow, which has already been cleaned. I have told you in my previous lecture, in previous video, that how you can clean the surface of the laminar flow. So after cleaning, we will place our sample on the sterilized paper which has already been autoclaved. We will turn the spirit lamp on. We'll place 3 Petri plates on the top, and one more patty plate for the surface disinfectant. 3 patty plates. We will pour sterilized distilled water. And in the first patty plate, we will add 2% solution of sodium hypochlorite. This sodium hypochlorite solution, it is 12% volume by volume. We can prepare 2% solution of sodium hypochlorite by adding 1.6% sodium hypochlorite in 8.4 ml of water by using the formula C1V1 is equal to C2V2, where C1 is the concentration of this solution and the volume, how much volume of the sodium hypochlorite is to be added in the 10 ml in order to prepare the final volume 10 ml solution, 2% sodium hypochlorite solution. So we will add 1.6 ml sodium hypochlorite, 12% in 8.4 ml distilled water to make 10 ml sodium hypochlorite solution. So you can use for this is in this case these are the tender leaf. If you want to sterilize, why do we sterilize these plant material? There are many surface borne pathogens present on the surface of the leaf which cannot be washed off even if we wash it into the tap water, because we are interested in the pathogen or the microbe which is deep seated into the tissues of the host plant for the tenders. So that is why we use this surface disinfectant. It is commonly known as the commercial bleach. So for the leaves we generally use 2% solution, but if the leaves are a bit tender, they are very small, we may use 1% solution. And for the twigs or the branches which are a bit harder, which are a bit tougher, we can use 3% solution. So according to your sample, we can change the concentration of our surface disinfectant. So we will add in the first plate, we will add 10% sodium hypochlorite solution, which we have already prepared, and I told you the procedure how you can prepare 2% solution. Now we will take the scissors and we will cut the sample . We will sterilize the scissors on the flame till it get red hot. Let it get cool. Give it a dipping in the methylated spirit so that we may increase the process of cooling. Let's wait, be patient. Cut your sample along with the leaf spot, along with some healthier part. Why? Because the pathogen is generally progressing towards the healthier part. So along with the disease part, we will also take the healthier part of the specimen. Again, do it red hot. Sterilize it. Give it a dipping. Let it get cool. Wait a minute. You may cut each spot into 4 parts. Give it a dipping for about 30 seconds to 1 minute. With the help of the pointed forceps. Transfer your sample for the washing in sterilized distilled water. Give them 3 washings similarly. Transfer it into the third battery plate containing sterilized distilled water. So in this way we will wash them thrice in the sterilized distilled water. Now we will take the filter paper and we will place it on the filter paper which we have sterilized or autoclaved, and we will place it over here. Again we will sterilize our forceps. We wait, and after giving the third washing we will pick up our specimen, our sample from which we are going to isolate the pathogen which is responsible for causing the disease. Let the specimens get dry completely. We will wait till these samples are dried. We will again sterilize our hands and sterilize our top again. By the time our specimen is getting dried, we'll cut the paraffin. By using this paraffin, we seal the battery plates. We will cut it into the small pieces. This is how. I will tell you later on that how we will use this paraffin to seal the plates, so that the mites or other contaminants may not enter into the battery plates, even if they are placed in. Because when we place it in the incubator, there are many other cultures placed by many people at the same temperature. So one culture may contaminate the other. That's why we have to seal the battery plates. I think our specimens, they have been dried or about to dry. This is the time you have to be very patient, because if we let the moisture be there in your sample, they may enhance the growth of bacteria. Bacteria is bit fast growing as compared to the fungal growth. When you inoculate in your media, the very next day it may fill with the bacterial colonies. So you have to be very patient to get your samples get dried. I think they have been completely dried. So before inoculating them, we will label the battery plates. First of all, we will write the date of inoculation. We will write the date of inoculation, and we will write our sample number, R1. And we'll pass it over the flame, so that if there is any contaminant or fungal spore or any bacteria, it may die. Now, with the help of our thumb and the forefinger, we will lift the lid up on the top of the flame, and after sterilizing and cooling your forceps, we will pick up the specimen, the first bit of the leaf, and place it on the media. Again, we will sterilize it. We will turn the patty plate and pick up another bit and place it on the media. Again, we will sterilize it. Wait for this forceps to get cooled down. Hold the next specimen with the help of the forceps. Turn the plate. Lift the lid up and put your next bit in the next corner. Again, sterilize. The tip of the forceps. Let it get cool. Hold the specimen with the help of the tip of the forceps and place it on the surface of the media plate. So before putting it back, sterilize it, give it a dipping, and place it in the tool tray. So this is how you placed 4 specimens. You can place 4 specimens at a time for the isolation of the deep-seated pathogen in the sample, which is the cause of the disease. So we will place this plate, we will wrap this plate after inoculation. This is how you tear this paper. Hold it like this with your 2 fingers. With the help of these 2 fingers, hold the edge of the parafilm on the open ends. Pull the tape with the help of your right hand. And turn it all around the battery plate. So you can see this plate, it has been sealed by using this paraffin. Now we will place this inoculated plate, which has already been labeled, into the incubator at 25 degrees centigrade. After about 48 hours, we will be able to find the small hyphal growth coming out of these specimens, and we can pick these protruding hyphae from the edge of the these specimens and we can place it on fresh agar plate for the purification of the fungal culture. So this is how we can get the pure culture of the fungi. Now I'm going to place this plate in the incubator at 25 plus minus 2 degrees centigrades.